A very good evening to everyone. Today we are going to continue our question practices. As you know, yesterday we have started by with the practice of previous year questions, and yesterday we have completed the first chapter, right? First chapter of genetics, that is principle of inheritance. So today we are going to complete and discuss some of the previous year questions of the chapter number 6 of your 12th class, that is molecular basis of inheritance. Okay, so before starting the session, please let me know through this technical poll whether I am audible and visible to you or not. So here is the poll. You have to give your answer whether I am audible and visible to you clearly or not. Okay, so everything is fine. Okay, so let's start with our first question. So our first question here given is, which one of the following sentence about the histones is wrong, right? So here the question is asking about the histone proteins. You know, the histone proteins are very we can say important molecules for the condensation of the DNA, right? So here are some statement given regarding this histone, and we have to find out the wrong one. We are not going to find out the correct one, we have to find out the wrong one. So as you know, the 4 different statements are given, out of which 3 has to be right and 1 has to be wrong, right? So your option A says the histones are organized to form a unit of 8 molecules, right? Option B says the pH of histone is slightly acidic. Option C says the histones are rich in amino acid like glycine and arginine. And option D says the histone carries the positively charged in the site. Okay, so you have to give your answer through this MCQ poll. I'm launching the poll. Please give your answer through this poll. And I'm keeping this poll for the 20 seconds only. You have to find out your answer and feed it here. Okay, and those who are not able to answer in poll can give their answer in chat box. Oh, okay, so as the time is about to over. Okay, so here I have ended the poll. Okay, so most of you have opted for the option B, which says the pH of the histone is slightly acidic. As you know, histone is a protein which has to wrap around, or around which DNA has to wrap, right? If DNA has to wrap around this histone protein, so this histone protein has to be of opposite charge, right? Only then the linkage can be settled. We know the DNA, due to the presence of phosphate and hydroxyl group, the DNA is negatively charged. DNA is negatively charged, right? So the histones has to be the positive charge molecule. So according to that, your D is right, right? And this positive charge is only given by this positive charge which are rich in the histone protein is lysine and arginine. So the basic protein which we have is lysine and arginine, right? So according to that, third is clear, also correct. And when the DNA gets wrapped around the histone, these histone further encircle, start wrapping around themselves, start forming a coil, and in the coil they contain the 8 molecules. That is why it is known as histone octamer, right? If the histone octamer is formed, so the option A is also correct. So now we can say the option B, which is saying pH of histone is slightly acidic, is incorrect, because due to the presence of basic amino acid, it is forming a basic pH. Okay. So for the question number first, the answer is option number B. Okay, because it contains the basic proteins, that is lysine and arginine. Okay. Now we are moving towards our second question. The second question here is, which is the only enzyme? We are talking about the only, that means single enzyme that has capability, that has the ability to catalyze the initiation. It is catalyzing initiation, elongation, and the termination in the process of transcription in prokaryotes. Okay, so transcription is taking place in prokaryote, and transcription involves three steps: initiation, elongation, and termination. What is transcription? Transcription is the process of conversion of DNA to RNA, right? Transcription is a process of conversion of DNA to RNA, or we can say the RNA synthesis, right? So here we have the four options. A says DNA dependent DNA polymerase. B, DNA dependent RNA polymerase. C, DNA ligase. And D says DNase. So here is your poll. You have to give your answer through this poll. 20 seconds has to be given, right? We have to talk about the transcription, right? The formation of RNA from the DNA. And for that formation, we are talking about a single enzyme, which is responsible for all the three processes. That is initiation, elongation, and termination. Only 5 seconds are left. Okay. So let's check which option is correct. So the option A says DNA dependent DNA polymerase. DNA dependent DNA polymerase. That means here is the enzyme polymerase. Polymerase enzyme is always for the elongation or the synthesis of molecule. But the synthesis of doing of what? The DNA. Here DNA polymerase is present. So we can say here synthesis of DNA is taking place. But the process is transcription, where we have to make the RNA from the DNA, right? So option A cannot be the answer. Option C says DNA ligase. Ligase is to bind something, but we are not binding something here. So option C is also not correct. Option D says DNase. DNase is an enzyme which has a cleavaging property, right? It has a cleavaging property. It will break the DNA, right? So we don't want to break the DNA or fragmentize the DNA. We want to convert, or we want to make the RNA from the DNA. So here the template will be our DNA, and we have to make the RNA. The template is DNA and we have to make the RNA. So yes, we can say it is an enzyme which is given in option B, DNA dependent RNA polymerase, because the enzyme is going to read the DNA, but it will form the RNA. Okay, so the answer of question number second is B again. Okay, is it clear to everyone? Okay, great. So let's move to the third question. The third question here is asking, which of the following RNAs is not required for the synthesis of protein? Here, now we are going to talk about the process of translation. Okay, here we are talking about the process of translation, in which we have to convert, or we have to make RNA to protein. That means we have to go for the protein synthesis, right? Now, in this question, we have given the four options. Among these four options, we have to find out the which RNA is not capable of synthesizing the protein, right? We have to find out the RNA which is not able to synthesize the protein. So option A is mRNA. Okay, some misprinting is here. Option B is tRNA. Option C says rRNA, and option D is siRNA. Okay, so You can give your answer in chat box if you have done the question, or you can wait for the poll. So as you have already given the answer in the chat box and you have opted for option D, let's check it out. Okay, so option A is mRNA. We know that mRNA is our messenger RNA, which used to the synthesis of protein, which will further give rise to or formation of enzyme. The rRNA is known as ribosomal RNA. It is going to produce the ribosomal proteins, right? It is again forming a protein. Then we have the tRNA that is used to form the transcriptase RNA, that is tRNA, the clove-like structure, right? So only RNA which is not going to synthesize the protein is siRNA, because this small interference RNA are used to inhibit, they are used to inhibit, right? They are used to inhibit the synthesis of protein, right? So this process we have also studied in RNA interference. So we can say the answer for the question number 3 is option D. Okay, the answer is option D here. Now the next question for you is, if the adenine makes 30% of the DNA, if adenine is making 30% of the DNA molecule, what will be the percentage of thymine, guanine, and cytosine? Okay, these are the given four example options to you. Okay, we know that in DNA we have the 4 nitrogenous base present: A, T, G, and C. Okay, now give your answer in this poll. Which option is correct regarding the percentage of the thymine, guanine, and cytosine, where the percentage of adenine in a DNA is 30%? Okay. Okay, great. So everyone has given the answer, and you can see they have opted for the option C. Let's find out which option is correct. Okay. So we have well aware of the Chargaff rule, right? We are well aware of the Chargaff rule, and the Chargaff rule says A plus T plus G plus C is always making 100%, where AT or GC ratio is always 1. That means whatever amount of adenine is present, that is equal to the amount of thymine. Whatever amount of guanine is present, that is equals to the amount of cytosine, because A pair with T and G pair with C. If A is 30%, that means T is also 30%. So 30 plus 30 become 60%. 60% of DNA is carrying A and T molecule, right? T, A, and T nitrogenous bases. Now the rest 40% is G and C, the combination of G and C. So we can say G plus C is equals to 100 minus 60, that is 40%. If G plus C is 40% and G amount is equals to C amount, then we can say G is equals to 20%. So it is also equals to the C. So if we look at these options, only the option C is there, where the amount of thymine is 30, amount of guanine is 20, and amount of cytosine is also 20%. So yes, well done, you have opted a correct option. Okay, your option is correct, and the answer for this question number 4 is C option, right? So let's move to the next question. The next question says, complete the flow chart on the central dogma. We know the central dogma is always talk about the sequence. It is always talking about the sequence which is required for the formation of protein, right? It is a sequence which starts with the DNA and ends up with the protein in a sequential manner. So you have to find out the correct answer. Okay, so you have already given your answer in the chat box. Let's find out. We know the central dogma goes from DNA to RNA and RNA to protein, right? But before that, the DNA itself get replicated. So the first step is always replication, right? The making a copy of DNA. Second says the conversion of DNA to RNA, that is transcription, right? So it is transcription. The formation of protein from the RNA is known as translation, right? And the last, we have the protein molecule. Let's find out where these four steps are given. So in option A, it is given replication, transcription, transduction. Transduction is not there. Transduction is a process of transfer of DNA from one bacteria to another. It is a process of the gene transfer, not the central dogma process, right? So the translation is given in option C. Let's find out. Option A, sequence in A is replication, B, transcription, C, translation, and D, protein. Yeah, this is the correct sequence. So yes, the answer for the question number 5 is C option. Okay, it is absolutely right. Now, let's move to the next question. The next question says, E. coli has only 4.6 into 10 raised power 6 base pair. Here we have 4.6 into 10 raised power 6 base pair, and the E. coli has the ability to complete the process of replication within 18 minutes, right? It is having the capability to complete the process of the whole replication of DNA in 18 minutes. Then we have to find out the average rate of polymerization. We have to find out the average rate of polymerization. So find out, it is a numerical. Please calculate it and let me know the answer through this chat box, through this MCQ poll. So here is the MCQ poll, and it will be remain here for 25 seconds. You have to calculate the answer and feed your answer in the MCQ poll. That out of these four options, option A says 2000 base pair per second, option B says 3000 base pair per second, option C says 4000 base pair per second, and option D says 1000 base pair per second. So we have to find out what is the average rate of polymerization is present here. Okay, find out the answer. Okay, now I'm going to end the poll. Okay, so as you have opted for option number B, okay, so this is your result. Let's check out which option is correct, right? So as we have the base pair, that is 4.6 into 10 raised to power 6, right? And the time consumed here is 18 seconds, take 18 minutes. So we can divide it with seconds because we have to find out the answer in seconds. So 1 minute is equals to 16 seconds. So 18 into 60. So we can write 46 into 10 raised to power 5 divided by 0. 1080. So we have to divide 4, 6, 1, 2, 3, 4, 5 divided by 1080. So when we find out the answer, it will come around approximately 4000 base pairs per second. Okay, after division, we'll reach to the result that it is having the average rate of polymerization, that is 4000 base pair per second instead of 3000. Okay, so if you want to calculate, you can divide this, right? So the answer for this question is C, that is 4000 base pair per second. Okay, so the next question says, name the enzyme that facilitates the opening of DNA helix. We have to open the DNA helix during the transcription. Please keep this term in mind. We are talking about the opening of DNA during the transcription, right? Your option A, DNA helicase, option B, DNA polymerase, option C, RNA polymerase, and option D, DNA ligase. You have to give your answer through this poll, that which enzyme is responsible for the opening of DNA helix during the transcription. Okay, let me end the poll. Okay, as you can see, you have opted for option D. Let's check out which option is correct. Okay, here we are talking about the transcription. Transcription is a process in which DNA gets converted into RNA, right? So whatever enzyme is working here, it is always DNA dependent. But as the synthesis of RNA is taking place, so it is a RNA polymer. So DNA helicase works during the replication of DNA. When we, if you remember the fork-like structure which is formed during the semi-conservative nature, we have explained during the replication of DNA. So DNA helicase was working there. That is during the replication of DNA. So it cannot be the answer. DNA polymerase, as the enzyme is polymerase and the DNA polymerase is there, so it is causing the synthesis of DNA, but here we are not synthesizing the DNA. Next is the RNA polymerase. That means the synthesis of RNA. Let's see the last. It is DNA ligase. The work of ligase is to bind the DNA, right? But we are not binding something. So RNA polymerase, as you know, in prokaryotes, RNA polymerase is the only enzyme which is responsible for the opening of the DNA. When the RNA polymerase binds to the promoter region, it leads to the opening of DNA, right? And the synthesis of RNA. So instead of option D, the correct answer is option C, that is RNA polymerase. Is this clear? If is it clear, please use the thumbs up reaction. Okay, so now we are moving to the next question. Okay, so the next question says that who coined the term kinetin? Okay, this term is you have familiar with. So who is the scientist who have coined the term kinetin? You have to find out. So this kinetin term is given for by which scientist? Your option A, Miller, option B, Darwin, option C, Venn, and option D is Kurosawa. Okay, so here is your poll. You have to give your answer. 20 seconds has to be given. Okay, within 20 seconds or after 20 seconds, I will be ending this poll. Okay. So here is your result that you have opted for option this. Let's find out. Okay, the kinetin is a term used for an hormone. It is an term used for an hormone which is found in plants. Okay, which is commonly known as cytokinin. Okay, it is commonly known as cytokinin. In the growth regulator chapter, in the plant growth regulator chapter, we have seen the kinetin term was given by the Skoog and Miller in 1955. In 1955, and this kinetin is also known as 6-furfuryl aminopurine. This kinetin is also known as 6-furfuryl aminopurine, and initially this kinetin, that is cytokinin hormone, was extracted from the herring's sperm. Okay, it is extracted from the herring's sperm. So the correct answer for this question is option E. Okay, is it clear? Okay, So let's go to the next question. Our next question says, in the polynucleotide chain of DNA, a nitrogenous base is linked to the OH group of the hydroxyl group of. हमारे पास we know that DNA is a polynucleotide chain, that is made up by the joining several nucleotide, okay, molecules. So when the nucleotide is formed, the nucleotide, the nitrogenous base always get linked to a sugar, right? It always get linked to the sugar. So the question is asking, to which hydroxyl group of which carbon of the sugar is this binding takes place? Your options are given here. A, 2' carbon pentose sugar, 3' carbon pentose sugar, 5' carbon pentose sugar, and the D is 1' carbon pentose sugar. Okay, so as you have already given your answer in the chat box, let's see. You know it is a DNA. So in DNA, we are using a pentose sugar. That is a deoxyribose sugar, we know, because we have converted the hydroxyl group at the second carbon into the hydrogen. And on the first carbon, we have the hydroxyl group, right? So there is a fifth carbon, H2, CH2OH. So first carbon, second carbon, third, fourth, and the fifth, right? When we look at this, on the fifth carbon, the phosphate moiety binds, right? So the first carbon is the left. Here, the hydroxyl group of first carbon binds with our nitrogenous base, right? So it is a 1' carbon pentose sugar. So according to that, option B is a correct answer. Okay. Okay, so next question is, the term nuclein for the genetic material was used by. The term is nuclein. It was first used by which scientist? Your option A, Franklin, B, Mesher, C, Chargaff, and D, Mandel. So here is your poll. You have to give your answer in this poll. Again, 20 seconds has been given to you. So you have to find out the name of the scientist who used the term nuclein, right? Option A is Franklin, Mesher, Chargaff, and the Mendel. Okay. So as you have given the answer, okay. So now I'm ending the poll. This is your opted option. Okay, let's find out. The Mendel, okay, the Franklin, Chargaff are related to the discovery of DNA, right? Mendel is related to the description of the behavior of gene, the nature of gene, and the function of gene. So Frederick Mesher is the scientist who has used the term nuclein, and this term nuclein is used for the nucleus of the pus cells, right? It is used by the scientist Frederick Mesher, and he used this term for the nucleus of the pus cell. So the correct answer is option B, that is Mesher. Next question says, which of the following statement is correct? Now you have to find out the correct statement. Okay, that means out of these three statements are incorrect. You have to find out the correct one. Option A says adenine pairs with thymine through one hydrogen bond. Option B says adenine pairs with thymine through the 3 hydrogen bonds. Option C says, okay, adenine does not pair with thymine. And option D says adenine pairs with the thymine through the 2 hydrogen bonds. As we have already discussed, we have the 4 nitrogenous bases A, T, G, and C, where A binds with T and G binds with C. And for the binding of these nitrogenous bases, we use hydrogen bond. So for the G and C, we require 3 hydrogen bond, but whereas for the AT binding, we require 2 hydrogen bond. And as you have opted for option D, you have given your answer option D. So let's find out what the statement of option D says. The adenine pairs with thymine through the two hydrogen bonds. Yes, so you have given a correct answer that adenine always pairs with the thymine through the two hydrogen bonds. Okay. Okay, now here is the next question. Next question is saying, if the distance between the two consecutive base pairs, how many base pairs? Two consecutive. That means they are present simultaneously, adjacent to each other, is 0.34 nanometer. This is the base pair and this is the base pair. And the distance between these base pair is 0.34 nanometer. And the total number of base pair of the DNA double helix is a typical mammalian cell. We are talking about the mammalian cell. So in mammalian cell, we have 6.6 into 10 raised power 9 base pairs. So you have to find out the length of the DNA, right? The number of base pairs are given and the distance between the two consecutive base pair is also given. Okay, the answer given here in the option is in meters. So let's find out. Okay, you have given your answer in chat box. Let's find out it is correct or not. So initially we have to convert this distance into the meter. So as it is given in nanometer, so we have to convert to 0.34 into 10 raised to power minus 9 meter, right? It is the distance between the two consecutive base pair, and here we have 6.6 into 10 raised to power 9 base pair. So we have to multiply this with the 0.34 into 10 raised to power minus 9. So this value cuts with this because one is positive, one is negative. So we have to go for the multiplication of 6.6 into 0.34. So when we done, it is also given in our NCERT, the answer comes up around 2.2 meters. So yes, you have opted a correct answer. The answer is option B, that is 2.2 meters. Okay, well done. So let's move to the next question on that behalf. In RNAi is the technique, that is RNA interference, right? It is the technique RNA interference. The genes are silent using. In this technique, we are using a particular type of RNA for silencing the gene. So what type of RNA we are using? Option A, double-stranded RNA. B, single-stranded DNA. C, single-stranded RNA. Or D, double-stranded DNA. You have to find your answer and okay, sorry, you have to find your answer and lodge the answer in your this poll. Okay, so you have already launched your answer. Okay, so let me end the poll. As you can see, you have opted for option A, right? You have opted for option A. Okay. So yes, in RNAi, that is RNA interference technique, which we have used for the pest control method, right? We have used this technique in the tobacco, against the tobacco plants, right? In the tobacco plant, there is a pathogen, which is a nematode present in the roots of the tobacco plant, which is hindering the growth or the yield of the tobacco. And to resist the functioning of protein of that nematode, we have used the double-stranded RNA. Yes, you have given a absolutely correct answer. That is double-stranded RNA. Okay. Next question for today is, what initiation and termination factors are involved in transcription in eukaryotes. Okay, question आपसे पूछ रहा है that which factor of initiation and termination are required in the eukaryotes. Option A says sigma and rho, B says alpha and beta. Okay, there is some mistake in this question. These options are not given for the eukaryotes, it is given for the prokaryote, right? These factors are present in prokaryotes, right? So we have to find out which is the initiation and termination factor. Okay, so the answer for this question is option A, the sigma and the rho factor. This sigma factor binds to the promoter region, right, to initiate the polymerization, or sorry, the transcription, or we can say the polymerization of RNA, and rho factor is used for the termination. By binding to the rho factor, the termination, the polymerization ends. Okay, so in prokaryotes we have the factors sigma and the rho. If we talk about the eukaryotes, there we have the another factor, that is transcription factor 2A or the termination factor 2H. Okay, these were the factors required for the eukaryotes. But here the misprinting is there in question, so it is not for eukaryote, it is for prokaryotes. So in prokaryote we have two factors, sigma and rho. So option A is correct. Next question here is, which scientist experimentally proved that the DNA is the sole genetic material in bacteriophage? Here we have to find out that which scientist has says that DNA is the genetic material in bacteriophage. Okay. Okay, so you have already given your answer before reading the option. So you have opted for option C, that is Hershey and Chase. Yes, Hershey and Chase are the scientists who has used a bacteriophage. You know, they have cultured those bacteriophage in two different medium, the radioactive labeled phosphorus and the radio labeled sulfur. When they infected the bacterial cells, that is E. coli, with these bacteriophage, then they got to know that the radio labeled phosphate group, that is which is present in the nucleic acid inside the proteinaceous code of bacteriophage, is the one who enters inside the bacteria and causes the infection. So yes, option C is correct. Okay. Next question 16 is saying, what will be the sequence of mRNA produced by the following stretch of DNA? Okay, here two strands are given of DNA. One is template strand and another is coding strand. You have to find out which strand is complementary, which strand of RNA is complementary to the strand of DNA. As we know, during the transcription, the template strand is used for the synthesis of RNA. So you have to give your answer through this poll, that which out of the given four options, which one is the correct option? Which sequence is correct of the complementary RNA? Okay, great. So you have lost your, you have given your answer and you have opted for option C. Okay, so let's see, is it correct or not? So our template stand is having A, T, G, C, A, T, G, C, A, T, G, C, A, T, and G. As you know, in DNA we have thymine, whereas in RNA we have the uracil. So the complementary to A is always uracil, and to the thymine we have adenine. Okay, only difference between the DNA and RNA is that the replacement of thymine with the uracil. So U, A, C, G, T, sorry, U, A, C, G, U, UACG. As you can see, UACG is repeating itself again and again. So we have to look for that. So only the one strand is there which is starting with the U. That is the third one. So yes, third option is option C is correct, right? Okay, next move to our next question. Next question says, which of the following nucleic acid is present in an organism having 70S ribosomes only? Okay, your option A is single-stranded DNA with protein code. Option B, double-stranded circular naked DNA. Option C, double-stranded DNA enclosed in nuclear membrane. And option D is double-stranded circular DNA with histone protein. Now you have to give your answer through this poll. I'm launching the poll. So here is the poll and it is for the 20 seconds. Please give your answer. Okay, so when we are talking about, when we discuss about the 70S RNA type, right? 70S ribosome, we are always considering the prokaryotes. We are always considering the prokaryotes, right? We have two type of ribosome, 70S and 80S. So 70S is for the prokaryotes. So we have to find out the nucleic acid condition regarding to the prokaryotes. Okay, you have given your answer. So this is the option which you have opted. Let's find out which option is correct. As you know, the bacteria is itself a prokaryote, right? It has a chromosomal DNA, right? Which is a double-stranded linear DNA, which is a double-stranded linear DNA, which is present in a nucleus, right? Because there we do not have any membrane-bound structure of nucleus, and it does not contain any histone protein. So it does not contain any histone protein. So option D is wrong. Along with these linear, along with this linear double-stranded chromosomal protein, we have some double-stranded circular naked DNA also present in the cytoplasm. And these circular double-stranded naked DNAs are known as usually the plasmid in the biotechnology, right? So your option A was single-stranded DNA with protein code. No, there is no protein code present. Option A cancel. Option D also get cancelled. Option B says we have the double-stranded circular naked DNA. Yes, we have the double-stranded circular naked DNA present, but it is not present in chromosome. It is an extra chromosomal DNA which is present in the cytoplasm. So option B is correct. Option C say it is having enclosed in a nuclear membrane. No, because prokaryotes are only have the feature that it is not having any nuclear membrane. So option C is also incorrect. So correct answer for this question number 17 is option B, that the prokaryotes contain the double-stranded circular naked DNA. Okay. Now, next question. Question number 18. It is saying purines found both in DNA and RNA. Purines, that means here we are talking about the nitrogenous bases. Option A says adenine and guanine. Option B says guanine and cytosine. Option C says cytosine and thymine. Option D says adenine and thymine. You have to give your answer through this poll, and poll will stay here for 20 seconds. So opt for your option carefully, that here we are talking about the purine kind of nitrogenous base. You know, we have two categories of nitrogenous base. We have two categories of nitrogenous base. One is purines and another is pyrimidines, right? And in total, we have total 5 types of nitrogenous bases. Okay, so as you have given your answer in the poll, and this is your result, you are saying that option B is correct. Okay, let's find out. Option A is correct. So let's find out. Option A is saying adenine and guanine. So as I told you, nitrogenous bases are present in two categories, right? Purines and pyrimidines. The purines have two nitrogenous bases, adenine and guanine. that is A and G. Yes, this is present in DNA as well as RNA. Whereas in pyrimidines, we have three pyrimidines, that is uracil, thymine, and cytosine, out of which two are present in DNA, whereas the rest two are present in RNA. So in this question, we are talking about only purines. So yes, your option is correct. Option A is the correct answer because the purine which we have, the adenine guanine, is present in the both type of nucleic acids. Correct. Okay, so the next question, I think it is the last second question because today we are going to complete only the 20 questions. So this question says the first, the experimental proof for the semi-conservative replication of the DNA was shown in which experimental model? Option A says plant, B says bacteria, C fungus, and D virus. So you have to give your answer through this poll. Okay, before launching the poll, you have already given your answer. Okay, in the chat box that you are looking for option B, that is bacterium. Yes, it is option B, the Hershey and Chase experiment, right? The semi-conservative replication of DNA was given by, sorry, it is not the Hershey and Chase experiment. It is given, okay, sorry. So the semi-conservative nature of DNA was shown in the bacterium in which we have uh, cultured our DNA in the isotopes of nitrogen. One is N14 and one is N15 to find out which layer of the DNA, which layer of the nitrogen isotope is formed after the centrifugation. So yes, option B is correct. Now, question number 20 and the last question of the day. The question says the final proof for DNA as the genetic material came from the experiment of the final proof which says that yes, DNA is the only genetic material came by the experiment of which scientist? A. Griffith, B. Hershey and Chase, C. Avery, McLeod, McCarthy, and D. The Hargobin Khurana. Okay, so you have given your answer in the chat box and you have opted for the option B, right? That is correct. The Hershey and Chase experiment. Okay, the Hershey and Chase experiment have given the final proof that DNA is the genetic material, and they have worked. They have worked on the using two organism. One is bacteriophage and another is bacteria, right? So it is a correct answer in which they usually labeled the nucleic acid of the bacteriophage with radio labeled phosphorus and the protein coat with the radio labeled sulfur, right? And when they infected the bacteria with the bacteriophage, they seen only the radio labeled phosphorus material, that is a nucleic acid, enters inside the bacteria and causing the infection. Yes, so option B is correct. So this is all for today and we have successfully completed all the questions. I hope this session helped you to understand the concept of this question, and you also get to know in which topics you have to work a little harder. Okay, so with this we are going to end our today's class. So here is the NPS poll. You can give your feedback, your precious feedback, through this NPS poll, right? And after giving your feedback, you can leave the class. So this NPS poll will exist here for 25 minutes. You have to give your feedback in this and then you can leave the class.